Welcome, and it's great to introduce today's speaker, Anup from the Institute of Genome Sciences. Anup is wearing a lot of hats over there. He's the Chief Information Officer. He's also Executive Director for Software Engineering and IT, and Director of the Bioinformatics Core. And he's also the Executive Director for Software Infrastructure at the Institute of Health Computing. Anup has an undergraduate degree in Electronics and Communication Engineering and a master's degree in International Management. And he has been working in biomedical research for more than 30 years. He has a very impressive CV, a long string of collaborative publications, and he has also been on numerous grants, well-funded over the years. At the Institute of Genome Sciences, he's overseeing the bioinformatics analyses, the IT infrastructure, and he develops software tools for genome sequence analysis. And he's also responsible for developing data visualization tools used in clinical and genomic research. It's great to have you here, and please take it away.
Thank you, Dr. Benson and Dr. Mazzino, for inviting me to give the grand rounds, and it's a pleasure. And I apologize in advance for people on the Zoom. If you're on a small screen, some of the slides might be a little hard to read, but happy to sort of talk you through it. So my talk today is about accelerating scientific discovery and precision medicine through improved data accessibility and use. And so what that really means is that essentially the tools we build are trying to make data more accessible to non-data scientists to make it more accessible data. So I've got 4 topics that I've created slides for, and depending on how our conversation goes, I may or may not be able to cover all of them, but I'm hoping to at least get into the first 2, which is the probing outcomes data with visual analytics and the gene expression analysis resource, because those I think are more pertinent to this group and this division and this. And if time permits, then we can talk about the next 2 topics. Already introduced, currently sort of co-lead the Informatics Resource Center with Owen White and Dr. Michelle Giglio. And our team is made up of about 12 engineers, 3 computational biologists, about 7 bioinformatics analysts, and about 4 IT staff. And what I'm going to present is a collective work from all this group. So, that's where the credit really belongs. I just sort of help along to split over these things. So the first topic I'll cover is called probing outcomes data through visual analytics. And it's my pleasure to actually see Anne here because she's been with us for most of the journey in development of Partners. And so I really want to sort of acknowledge all the contributions. And as I mentioned, I'm going to sort of show the acknowledgement slide at the beginning because this is a team effort and sometimes that goes without saying. And I've highlighted a couple of them. In this particular case, Dr. Lisa Schulman, who's the main motivator behind this tool that we built about 7 years ago. She came to me with a prototype and she said, Anup, I heard that your team does web development. Can you help me build this tool where I want to be able to visualize my own clinical data? And that's the genesis of this project. And Jonathan Crabtree, who I've highlighted, is a senior engineer in my group, and he's the one who's been instrumental in architecting and helping us build the system. And along with the way, we've had a pretty sizable team helping us, people at University of Maryland, Dr. Michael Cummings and Dr. Nicholas Elmquist, who are both at CBCB and at the Human Computer Interaction Lab, have helped us build this tool. And then there are, as I mentioned, a few people from EPI, which is Dr. Ruber Baldini and Michelle Chardel, who's at IGS, who helped us along the way with some of the statistical aspects of the tool. As I mentioned, this was developed in collaboration with Dr. Lisa Schulman. The main idea is that we are trying to visualize and analyze phenotypic data. And I've mentioned that because when I talk to the crowded genomic data, when we talk about data, it's mostly genomic data. So in this case, I'm just clarifying that this is phenotypic data. And the idea that this is an easy to use tool for clinicians and for the non-data scientists. And the focus here is on hypothesis generation and confirmation and not advanced statistical analysis, because the idea is that clinicians can ask some questions, and once they have a hunch, they could come and talk to people like you to help them actually do the analysis. And so this is a web-based platform, so to make it accessible to people around the world. And as I mentioned, we've been sort of developing it for about 7 years. And currently we are at the stage where we feel proud about where the tool is. And we just started beta testing with a couple of locations. One is within UMB, And there's another instance of actually running PARTWIZ at the National Institute for Deafness and Communication Disorders. And the reason being that Dr. Rona Horsano, who used to be at UMB, is now moved to NIDCD, and she's been championing the use of this tool at NIDCD. So we have an instance running there as well. So the main features is that essentially it's a flexible enough tool to support disparate clinical data sets. And currently what we have loaded into PARTWIZ are a Parkinson's disease data set, and NHANES, which is a survey data set. And we also have some cancer data in there. And I'm going to sort of give you examples of Parkinson's disease data set as well as the NHANES data set today. And others are there if you want to talk about it. And so the main feature is that you can take a data set and either subset it by choosing one or more variables to include in your analysis, or you can also do a meta-analysis by combining more than one data set to increase your population size or to bring in disparate data sets together. And currently we support both cross-sectional analysis or data set as well as longitudinal data sets. And the main function is to essentially take these data and you create cohorts and you compare outcome variables for these cohorts to see how they're different between your cohorts. So these are then a visual representation of the main steps. So from the home page, you sort of first thing you do is select a data set through the data set selector, and then you create study groups. I use cohorts. Dr. Lisa Schulman likes to call them as study groups. So we sort of use those interchangeably, but they mean the same thing. And then once you've created your cohorts, then that's when you go to data analytics and you analyze the data. The first example I'm going to use is a Parkinson's Progressive Markers Initiative dataset. And so this dataset was a dataset here, which has been sponsored by the Michael J. Fox Foundation. And for people who don't really know, he actually suffers from Parkinson's disease, and that's why he's kind of funded this research over decades. So this is a longitudinal observational study, and the main idea is to sort of identify biomarkers that can sort of give a sense about the progression of Parkinson's disease and the effect on individuals. And there are about 4,000 volunteers in this particular study with spread across multiple cohorts, and the study was launched in 2010. So they almost have somewhere between 10 to 12 years of follow-up right now under this grant. So the first thing you do in PARTWIZ is once you have a data set loaded, and for loading the data set, we actually have currently at least you have to work with us because we take that source data set and we transform it into a form that we can load into PARTWIZ, and that's what we load, and that's kind of where we come in. And once the data set is loaded, you can sort of look at the information about the data set. So this is the information screen, and as you can see behind, there's a little description of the data set. And what I'm showing you is essentially you can take any variable and look at its distribution in your data set. So in this case, I've chosen the age at enrollment in the study. And this is showing you just a histogram of the data. And behind that, you can see that you've got a description of a pretty lengthy description of each of your variables. And hidden here behind your screen. And hopefully people can see my mouse on the Zoom. This is like a little, what I call as a sunburst chart. And that gives a distribution of what are the males and females in your data set. And you can expand it and you can even see the racial distribution within your data set. So quickly you can get a summary of your data set to see if this is of interest, what are the variables that you're working with. So you can sort of do the analysis based on that. So once you have the data set, the next screen is what I call as creating your study. So what you're doing here is essentially choosing to include one or two of your data sets to include. And here I'm showing you a meta-analysis. So the way PPMI data are divided is they have different cohorts. So there's a Parkinson's disease cohort and there's a healthy control. So these are two cohorts that are being followed. And what this is showing you is on the right here is the healthy control. And there are 228 subjects in that cohort. And here's the Parkinson's disease cohort with about 784 subjects in there. So what I'm doing is including both of these data sets to analyze them. And then it shows you all the shared variables between these two data sets so you can decide which variables to include in your meta-analysis in this case. And so essentially when I select each variable, it essentially either increases or decreases the number of subjects that are there depending on whether that variable has been measured for those individuals or not. And these little distributions is what we call as spark lines. And that sort of gives you a sense of what the data looks like. Just quickly looking at it, you can eyeball it and say, yeah, this looks like an interesting variable to include in my data set, or really I don't see any difference, and so I'm not going to include it. For instance, if you look at scope of AUT, you can see that actually there's a slight difference in distribution because that's actually one of the measures that's different for individuals with Parkinson's disease and not. But if you look at something like MoCA, further down here, really the distribution looks very similar. And you can actually, you'll see in later analysis that MoCA, there's no difference between individuals or significant difference between individuals with Parkinson's and without.
But this is longitudinal data, so there's multiple measures.
Absolutely.
So how is that being reflected?
I'll just show you in the next screen. Let's look at that. So this screen essentially allows you to look at sort of high-level distribution of the variables and decide which variables to include. And what you'll see at the top here is essentially a tracker. So because to make it simple, we have a tracker almost like, we've all used to start looking at trackers on website, which tell you where exactly are you in your analysis, and you can go back and forth between it. So essentially, the first one is create study data set, second is select study groups, and third is data analytics. And within that, these are the sub-steps which we have numbered them. So you can sort of go back and forth between these sort of screens. So here's where you can actually then start seeing for a longitudinal data set, how are your variables and how many measurements do you have at each time. So what you're seeing along the x-axis here are visits, and these are categorical visits, and the way PPMI measures the data sets is for healthy controls, they bring you in once a year to get the measurements, and for people who already they know are at risk or already have Parkinson's disease, they measure it every 6 months. And so essentially what you see these alternating bubbles is that for healthy controls, there's no measurement at some of these visits, and that's why the bubbles are smaller. And the bubbles are indicative of how large is your data set at that visit. For instance, at V01, which is visit 1, you can see the bubble is smaller than BL, which is baseline. So there's a legend here on the right, and the size of the bubble tells you how many subjects have that measurement at that visit.
Anup, I'm not sure the mouse is appearing on our screen. Can you just try to move it around so it would?
Oh, sorry, I was screwing it up here.
Yeah. Okay, there we go.
Yeah.
All right.
So this is the legend, and the size of the bubble tells you how many subjects are at each of the visits. So for instance, in the beginning at the baseline, the circle is bigger because you've got a lot of people who've measurements at baseline, and as time goes along, at visit 16, that you can start seeing the circle get smaller because you have fewer and fewer individuals with measurements for that particular variable at that time. So in this screen, in a longitudinal data set, you're trying to sort of set what is your baseline and what is your end of your study. And in this case, this green box that you see there is my baseline. And as you can see, that one measurement, which is MoCA, wasn't measured at baseline. So I've moved that over. So I'm taking the MoCA measurements at screening and rest of the measurements at baseline. And that's what the green boxes are showing you. And then the red boxes are essentially showing you what is the last visit that I'm going to include in my study. In this case, it's V12, which happens to be about 5 years follow-up. So essentially, if you look here on the top, it's showing you what is the average follow-up time, which is 5.1 years for this particular cohort. So once you have selected your visits and you've set your baseline and your final visit for your study, the next screen is where you actually go and choose a set of predictors and choose a set of outcomes to sort of filter your predictors to create your cohorts. So on the top, what I'm showing you on the top here is essentially here. So this is my predictors panel, and I've actually added 3 predictors. One is the age of enrollment, your new data set, which essentially is a proxy for telling me whether it's healthy controls or whether these are individuals with Parkinson's disease. And I also have sex as my third predictor. And each of these graphs are interactive and they all link together. So when I click on one graph, it updates itself. So in this case, what I've done is I've clicked on the healthy control, and that's why you see it as dark blue. And so essentially, dark blue is your filtered cohort that you're creating, and the light blue is the remainder of the population. And that's kind of what this is showing you. So in this case, because I've selected healthy controls, you can see at the age of enrollment, the dark blue is giving you a distribution of your individuals in your selected control, and the background light blue is the remainder of the population. So this gives you some feedback about what does the distribution look like for your selected cohort versus the rest of the population. And the second thing that happens in the bottom, you'll see is that essentially I've included 3 outcome variables. One is the ESS state, which measures the sleepiness, and we have MoCA, which is a cognitive assessment, and MDS-UPDRS3, which is a measure, which sort of overall measure for sort of change in motor functions and other functions in Parkinson's disease data sets. And along the y-axis, so x-axis here is, the first axis here is my baseline measurement, and the second axis on the y-axis here is the visit 12. On the x-axis, and on the y-axis is actually showing you the measure. In this case, it is MD-ESU-PDRS3 and MoCA and then ESS state, and giving you the proportion. So this is what the outcome variables panel does. It sort of shows you what is the difference in distribution. And if you look a little closely here, there are these little two blue dots. There's a darker dot and there's a lighter dot. The lighter dot again is the mean for the rest of the population, and the darker dot is the value for your selected cohort. So there again, we're giving you graphical feedback about, when you're selecting cohorts, how are your variables different between these two groups to decide whether you want to actually save that cohort or let it go. And once you have created a cohort, you can actually save it. And essentially the top right box here is showing you all the cohorts that have saved. And it's kind of small to read, and you'll see it on the next screen. But that's kind of what this is showing you, is all your saved cohorts in your analysis. And the bottom right here is essentially a simple statistical test that we do for each of the outcome variables between your selected cohort and the remainder of the population. So essentially, this is showing you here that in your selected study, you have about 153 subjects, and the remainder of the population is about 302 subjects. So you can sort of start getting some feedback through visual analytics right there, and you get some hints about the statistical importance of your cohorts that you're creating. So once you have created all the cohorts that you need, the next step is for you to go and actually run some simple data analytics by comparing these cohorts. And this is kind of what our data analytics screen looks like. So on top, we show you all the cohorts that you have, and there's a color for each of your cohorts, and the size tells you what are the number of individuals in your cohort.
And move your mouse back over.
Oh, sorry. Yeah. So here's the number here, which shows you the number of subjects in your cohort. And on the right-hand side, we give a textured description of what is the filter that was applied. So in this case, it's healthy controls and females is the first column. The second one is just the healthy controls. And the third is the data set, which is individuals with Parkinson's disease. So you can sort of select all of your cohorts and then choose one or more of your cohorts and select analysis. And when you select analysis, you've got three options. You can either look at your baseline variables for your analysis, or you can look at your last visit. In this particular case, I've chosen the change. So the change is between your last visit and your first visit. How much is the change in your variable? And that's what I'm going to use to run my statistics. So below here, I'm showing you an example of two analyses I've set up. So the first one is where I'm looking for differences between males and females with Parkinson's disease. And as you can see, the p-value for all of these analyses is pretty, the range from 0.169 to about 0.293. So it's saying there's really not a whole lot going on in these outcome variables between males and females. So essentially, there's no big difference in these outcome variables. But if you come down to the second analysis that I have at the bottom, here the cohorts I've created are between individuals with Parkinson's disease or the ones who are in the healthy control group. And the pink is the selection that I've chosen. In this case, it's the MDS-UPDRS3. And it's again, I've used the change between the first visit and the last visit. As you can see in the graph here at the bottom, the purple box is the box for healthy controls, and the green box is the box for individuals with Parkinson's disease. So as you can see, there's a huge change in the MDS-UPDRS3 for individuals with Parkinson's disease compared to the healthy controls. So this is kind of a quick summary of analysis, and each of these analyses is saved. So you can come back to it in the future to sort of see what analysis you did. And as you can see, this is just giving you a summary of it. And if you want to actually look at the details of your cohorts and analysis, you can actually go to so there's these little buttons here that it's hard to see. One says 4 and one says like expandable buttons. So you can click on it and look at your analysis on a bigger screen, which is essentially what I'm going to show you in the next screen. Is when you expand the analysis, you can actually see a little bit more information. So in this case, I've got 4 panels in my analysis on the output. And the top left panel essentially is showing you the outcome variables. In this case, I've chosen MDS-UPDRS3. And the top right, it's showing you actually the longitudinal trend in your data. So the purple line is showing you the healthy controls. And as you can see that the value is low, starts low and stays low. And the one which is the dark green, that's the line for individuals with Parkinson's disease. And as you can see, they start off at a higher value than a baseline, and that is getting worse. In this case, the increasing value is getting worse. And so the dark green line is sort of increasing, showing you that over the 6 years of measurement, that particular measure has gotten worse for individuals with Parkinson's disease. And for people who actually want to look at the table 1 and table two kind of displays that are common in clinical papers. We have also included that. So if you see at the bottom left here, this is your sort of standard summary of results of table 2. And you can also look at table 1. So as you can see here, textually, if you want to look at the differences, it's hard for me to see. But you'll see that in the MDS UPDRS case, the average change in healthy control is 2.5 plus or minus 4, whereas the one in the Parkinson's disease is about 29.9 with plus or minus standard deviation of 13. So not only can you visually see the difference, but we also actually give enough information in tables so you can actually look at the distributions and see if it's real, meaningful, and what is the effect size. Hard for me to keep moving my mouse back and forth. I don't know you can advance it. So apologize. So then the next example I'm going to show you, so the previous data set was a longitudinal data set. And the next example I'm going to show you is the one of a cross-sectional data set. And this data we actually got from the National Health and Nutritional Examination Survey. So this is a survey conducted by CDC annually. And this is being done by almost over 25 or 30 years. And so typically what they do is they try to find 5,000 participants which are nationally representative across all the counties in the US. And so it's a pretty good national survey. And they measure demographic variables, socioeconomic variables. They have a dietary survey to ask about their dietary habits. They have other health-related questions. But then they also have some medical, dental, and psychological, physiological measurements, including lab tests. And then in this particular case, because of Dr. Rona Hartsana, who's an otolaryngologist, had an interest in hearing thresholds, we actually brought in the audiology measurements, in which in this case it was hearing thresholds. So we used two data sets to include to load into Podwiz. One is for the survey period of 2015 to 2016. And the second one is from 2017. Normally it would have been 2017 to 2018, but because of COVID, they actually did not get the full enrollment, so they extended it. So it's from 2017 to 2020 is the measurement period for the second data set. Okay. So I'm going to skip some of the earlier steps I showed, but essentially I'm just showing you all the complex cohorts that can be created. So in this case, the top cohort that I'm showing you here is one where I've included individuals between the age of 6 and 20 with a BMI measurement between 13 and 25 and sex as females. So you can create pretty complex intersecting cohorts based on intersections of variables that you have included in your data set.
So the cohorts are mutually exclusive, aren't
they? They are, but they don't have to be.
Oh.
So for instance, if you look below, we have one which says sex male and age 6 to 20.
Yeah.
And there could also be one where I've included the BMI. So now they've got overlapping distributions. So what happens is if you were to analyze these two together,
Yeah.
we give you a warning saying that there are some subjects that are shared between these two. And so your analysis may not be valid.
Yeah. Okay.
So we don't stop you, but we give you enough warning so that at least you know what's going on there. So this just shows you that you can create these complex cohorts. And I'm going to show you a couple of analysis. So what we've done is, as I said, we are creating hearing thresholds. So we actually customize the display to be able to show you hearing thresholds. So what you see on the top right panel here is actually what you would go see as an audiogram, which is what most audiologists are used to seeing. So on the x-axis are the different frequencies at which you've got the threshold measured, and the y-axis is the decibel at which your hearing sort of sounds are presented to you that you can detect. So the 0 is essentially at 0 decibels, but normally typically it's 10 decibels and above is what you want to see. And what is it showing you is that I've created two cohorts. So the one cohort is essentially individuals who have smoking history and alcohol use history. And the second cohort is the one which doesn't use alcohol or smoking based on the survey. And as you'll see, the purple or the dark blue line is the cohort who have no smoking or alcohol history, and the green one is with smoking and alcohol history. As you can see, at higher frequencies, their hearing threshold is higher. So that is indicative that individuals who have both smoking history and alcohol use have a slightly worse hearing than the individuals who do not. Now, when I first created this analysis, I was like, wow, this is interesting. So let me go back and look a little bit more carefully. So then when I went and looked at the actual table, in this case, the summary of results, one of the things that you'll notice is that it's kind of small to read, but in this cohort with individuals who have smoking and alcohol use, there's a slightly higher, higher incidence of the ratio of males to females is very different. So it seems like essentially females are the ones who have reported that they do not use alcohol as much and they haven't smoked as much. So there are about 80% females in that group compared to 20% males. Whereas in the other group, there's almost a 50-50 split. And it's known that men tend to sort of lose hearing more than women do, and we don't know why. So the question then becomes, is this, what you're seeing visually, is that real? Or do you need to sort of dig deeper into the analysis to see whether it's the sex that's influencing it, or whether it's just the smoking and alcohol history? So essentially, these are the kinds of cohorts you can create and at least start exploring these data and then sort of do more sophisticated analysis by coming and talking to people and statisticians. That's sort of the whole idea, is that this is a hypothesis generation tool. And I'm showing you another example here. In this case, what we have done is we have divided individuals based on their BMI, and we've essentially created 4 groups based on their 4 quartiles on BMI. And again, here again is showing that individuals whose BMI is higher tend to have worse hearing. And that's also a known fact, but again, there's so many confounding factors, because individuals with diabetes tend to have hearing loss, or individuals with some other conditions tend to have hearing loss. So the question is it just purely BMI, or BMI is just a proxy for some of these other conditions? But at least you can start visualizing it and asking yourself these questions to dig deeper into your data set. So this is sort of just a couple of examples I'm showing you there of data, cross-sectional data set with including NHANES. So I've shown you that we actually have built this tool. We can show you that we can look at longitudinal data and cross-sectional data set. And the question is, where do we want to go from here? So we have this tool currently in beta testing, and we're beginning to get some feedback from users about what they see, what are the gaps in terms of functions, and where would they like to see this tool go in the future. So one of the things that what people have said is they want to be able to see how does an individual's audiogram look in the context of a cohort. So what I'm showing you here is, here's essentially an audiogram. Actually, what you see is both on my screen. I see on the single display. So on the left-hand side is the audiogram of one individual, and there are 2 panels here. So the panel on the left is the audiogram for the standard measurements, and on the right-hand side, these are extended frequencies. So at NIDCD, for the clinical protocol, they actually have higher frequency measurements that are normally not done when you go into a clinic. So you're looking at essentially both the higher frequencies as well as typical frequencies in which the thresholds are measured. And on the right-hand side, what I'm showing you is, if you had the cohorts, and these are the cohorts that we created using NHANES, you can superimpose, or in this case, actually, we created a synthetic data set just to sort of visualize this information. So this is not real data. But if you had these cohorts, right? So what I'm showing you is how is the individual doing compared to the cohorts that you might have created in your data set. So imagine you have NHANES, which is a nationally representative sample, and I could have created these cohorts, and I can now show you how does an individual do in context of the cohorts. And for audiologists, what is interesting is all the published literature, and if you look at the hearing threshold, they're all normed about 20, 30 years ago on predominantly white males. But what people are beginning to find out is that females tend to have a very different hearing threshold profile and loss as you age. And similarly, race and the color of the pigment of your skin also apparently seems to have an effect on hearing. So individuals with darker skin tend to sort of lose hearing less often than individuals with lighter skin. There again, people don't know why. And we have actually been able to replicate that information in the enhanced data set. That when we create cohort based on age and sex and race, the Black females tend to have the least hearing loss, and white males tend to have the highest hearing loss.
But what is missing in that kind of display is some kind of dispersion measure, isn't it? Because you don't know how extreme that individual would be in the population.
Yeah, so actually I'm not showing you, but we can show error bars on this. So,
yeah, I would expect that you had like a color wash. So you had, say, from the 10th to the 90th percentile or something.
Yeah.
And then you could start to discuss if this is an extreme value.
Yeah, so we are working on these displays. Currently, we have ability where you can go in and show the error bars on it. And then, but the idea of showing like a graded,
it's
graded
more
one.
tolerance limits than error bars, really, isn't it?
Yeah, no, that's a great suggestion.
It's the population dispersion that you have, I guess, typically. I don't know.
Yeah, no, that's a great suggestion and definitely that's something that we need to work on. So that's kind of one request we got from individuals and we are kind of prototyping that work. The second request we have is essentially people saying, if we had a statistical model, so the alternative is for instance of the precision medicine, right? Once we start accumulating all these large data sets, people want to start saying, okay, if I had a model that can predict hearing loss or hearing threshold, how is my individual doing compared to the model that has been built? And so here's an example, actually, we collaborate with a group at University of Iowa, where they have a survey and what they actually, this is sort of partially published work from Wu and Dunn et al., where they gave individuals two qualities of hearing aids. And they have a survey which essentially, it's called EMA survey, which essentially tries to measure, see how are they performing in real world situations. And they got the feedback on that. And so we took that data and we built a model saying that how does the hearing aid do, quality of the hearing aid affect their perception of improvement in hearing. And so there are two different hearing aids and you can either turn off the noise cancellation or turn off or turn on the noise cancellation. And those are the four sort of distributions we are showing you. So we built a model based on the feedback that we got. And then we said, okay, here's an individual who shows up in the clinic and in these fields is essentially information for that particular individual. And so we said, how does that individual do compared to the model prediction? So the dark star that you see here on the display is that individual in each of these sort of categories. And so essentially what we're saying here is that the quality of the hearing aid doesn't seem to have a huge impact on the perception of people's perception about hearing aids. That was one of the findings from the study that came out. But this is just, again, an idea about how you could take this tool and sort of make it as a decision support tool, where if you had a model, you can sort of bring in the model into Pardvis and kind of just use the cohort that you have based on which you build the model to kind of display and see how is one individual doing compared to the rest of it.
So these displays, are they, do you write code to get a display like this or the audiograms that you showed us?
So,
Or is it all something that can be designed from the user interface?
so things like box plots and line plots, and these are all built into the display. And so any data set that's loaded, which is categorical, shows up as bar charts and continuous variables, so these are box plots. So those are all standard displays, but something like the audiogram, we have to custom build it just to have an audiogram display.
I think maybe what he's asking is how much could the user configure some of the way that the displays work? As opposed to counting on an engineer sitting down and.
Yeah, pretty much. If you need your own
Yeah.
engineer in the back end, so.
Yeah,
Yes.
no,
So say here you compare four different, I guess they are aspects of.
the hearing aid. So the two
quality,
qualities and what
whatever
you've
it is,
done.
Yeah,
No,
so you can generate that. You
Yeah.
can say, yes, I want four
Yeah.
displays, I want this, I want.
yeah,
that.
so all of that can be done through point and click by the clinician.
Okay.
We don't need to get
Alright.
involved.
Is that something you've programmed in?
Yeah.
Already?
Mhm.
So the choices of what you can do are set based on what you program?
And that's also programmed based on the type of variable that is being measured. So if it's categorical, we show a certain display. If it's like, for instance, right, one of the things is some people like box plot and some people like violin plots with the distribution, right? And there's debate about which conveys the information better. But you could, in theory, you will allow the user to flip between a box plot or a violin plot to see, the display. Those are the kinds of things we've already pre-programmed in there, and we give them control to do that. But we will not allow them to see a continuous variable as a categorical display of bar charts. They'll have to sort of dichotomize it before the data are loaded into the system. Right. So one of the things that we are exploring currently at IHC is this whole idea that we want, now that we are beginning to do the beta testing with this tool, we are trying to see whether we can have deploy this tool within School of Medicine with help from IHC. So the idea here being that data could come from Epic or data could come from RedCap, or we could also have third-party databases like, for instance, social determinants of health. All those data could be brought into a data mart, and then we could load it into Podviz, and people could build models, statistical models by working with folks in your department, or we could essentially have AI ML models built around these data, and then we can sort of create this ecosystem where Podviz becomes the visualization tool, where all of these data sort of come together and allow clinicians to interact with this data. So that's kind of my presentation about Podviz. I'll pause for a few minutes, see if people have any questions before I switch to the next topic.
Can we ask questions from Zoom?
Sure.
Yes, Joanne.
So I have a quick question. Can you transform continuous variables or categorize them within the context of this, or do you have to do that outside and then import the data?
That's a good question. Currently, it's the latter. So we essentially work with you to dichotomize the data. Let's say, for instance, you have a continuous measure, which is hearing loss, like as I showing you, mild, moderate, and severe, right? And so there are decibel ranges. And if you want to not have the hearing threshold as a continuous value, but you actually want to create these three classes, you have to do it beforehand and then load the data into the system, as of now.
And one other question real quick. Do the NHANES analyses take into account the sampling weights that you usually apply in NHANES?
It does not. This is just a trial data set we have loaded, and actually we will work with. So for this particular audiology data set, we have somebody who works with NHANES, and she's advising on us on correcting that before we actually start using it in real world. But that's a good question and a good point.
I think the vision is great. I think it's a great idea that as we have more and more data sets with more and more variables available, that you would like to sort of democratize the access to those data sets, that you don't necessarily need an expert to start doing queries in the data set. I think it's a balance though, isn't it? Because at some point it starts to become counterproductive just to play around with stuff, doesn't it?
Yeah.
I think this division into sets, I understand why it's there. I guess for many medically trained people, maybe that's an intuitive way of doing it. But that's also sort of an extreme way of slicing and dicing a data set compared to, say, a modeling approach or whatever, isn't it?
Yeah.
Yeah,
no, that's a balance, right?
It is a balance, isn't it? And when does this tool stop, and when do you export it into a more sort of professional modeling environment or whatever?
Yeah, so the way we did not talk about that. So one of the features we have in Podviz is, let's say, for instance, a clinician creates these cohorts.
Yeah.
We have an export function where if they can click export, and essentially you get all the tabular data that they can then bring to a statistician and say, these are my cohorts, so we already have it classified by the cohorts, and then they can run more sophisticated analysis in collaboration with the statisticians. So that was our vision, and I think Anne's been kind of helping us, trying to sort of say, yeah, this makes sense for you to build graphically, and here I think you should just leave it for statisticians to work on it, because...
Do you have a user group of people who are not involved in development?
Users?
A user group, a group of people who are not involved in developing.
So currently the NIDCD is that. So essentially the NIDCD is our first beta where they were not involved in the development of the tool so far, and they're serving as our users.
Yeah.
But having said that, Dr. Schulman, because she wants this tool to be very useful, actually ran a user group study about 2 or 3 years ago, which actually helped us improve the tool a lot. So she reached out to some, I believe, statisticians, including a couple from your division, and a few non-clinicians from outside UMB, to sort of get them to use the tool and give us feedback saying that this is what works, this doesn't work, make these changes. And so some of those have been incorporated and others we're still working on it.
Yeah, I have a comment, which is that these questions are incredibly relevant. Like it's very common for people to do UI development, and there's really kind of in a bubble. And the next material that he has, which is this gear project, what they've done is it's a web resource, et cetera, but they've held workshop after workshop after workshop at meetings.
Yeah.
And there's I think they've on the order of like 100 people in these workshops, and people are really sitting down and using the interface. So that's me teeing off. Maybe you could just cover a little bit of your
Yeah.
That's, yes, the user experience is a big deal, let's just say that.
So switching gears a little bit, so that was for clinical data visualization. I just want to sort of leave you with a sense of, how we use similar sort of tools and technologies for genomics. So GEAR stands for Gene Expression Analysis Resource. Again, this is a fruitful collaboration with Dr. Rona Hartsano, who now close to 12 years ago approached us when we were still in Biopark, saying that, hey, Anup, I'm like in those days, the reason I got involved with it is we were doing gene expression analysis for Dr. Hartsano. And she says, that's great, you've given me all the lists, but what I really like to do is be able to visualize my own data and play with that. And we looked around for tools and we could not find a tool that did what she wanted it to do. And so we wrote a grant and we got grant funding to actually build this tool, and that's what's called GEAR. And again, this is lots of people have been involved in it, but the two main ones I want to call out are Joshua Orvis, who's sort of the senior engineer in my group, who's been working on this for the last 11 or 12 years, and he's sort of the main engineer and architect behind this tool. And then we've got a bunch of engineers and analysts who have come along and helped. And then Rona Hartsano is the PI for the grant, and we've sort of collaborated, but other people also contributed, including Michelle Giglio and Setament and Carlo Colantini, who have actually used the tool and gotten some funding and added data sets for it.
If you
Move the slide, I'd
move
say.
the 5 minute over to extend it.
So 3.5 hours, yeah. I don't know why we even have a limitation.
Yeah.
Right.
So I've already talked about this. I will not spend time discussing this. I'll just give you a sense of what the tool does. So this is what the UI looks like. So the idea is you can take one or more RNA-seq data sets and load it into GEAR. And we have done the data loads for people, but this tool has been built so that people can take their own data sets and load it themselves. And we have tutorials on how to do it. So once you have data sets, there again, it's hard to compare two gene expression data sets to each other mathematically and statistically easily for an end user to use. And so we try to stay away from that. And what we have done is essentially we call them as a data set in a box. So each data set has its own box. And so we don't try to sort of compare those data sets. We let the users visually look at the information and make their conclusions without us trying to get involved, because we want to preserve how the analysis was done originally by the users. So in this case, there's a little text box here where you can type in the gene that you want to look at, gene or multiple genes, and then you sort of choose a data set collection that we have set up, and then you can look at that particular gene in that data set. So what I've done here is essentially I'm just giving you an example of what the display looks like. So in this particular case, POW4F3 is a transcription factor, and people are trying to sort of see what is the expression of the transcription factor across different data sets. So each of these boxes that you see with the purple heading is its own data set. So in the first case, it's a P1, a mouse or single cell RNA-seq data set measured at a P1 time point, and below is essentially a P0 time point and P2 time point. And what you see on the top here is essentially a single cell RNA-seq data set. This is also a single cell RNA-seq data set. And the big difference is one of these is done using the newer technologies where you don't sort the cells. And the one on the bottom right here is done where you actually do fact sorting of the cells. So you know exactly what cell is going into it when you do the RNA-seq analysis. And this is a cartoon of, I believe, the cochlea, which goes from apex. And essentially what you see here is this particular POW4F3 gene. It's hard to see here, but essentially there's a light to dark gradation of the color, because spatially that gene is expressed differently across the spatial dimension of your cochlea. And that's the kind of visualization you can do, where you can essentially see along the axis of your cochlea, how is that gene expression different, which you would not be able to just see numerically. So that's sort of one idea here, is that you can sort of create a cartoon and display these data.
How is the location coded? I understand what you're saying, but how is that information coded?
So each of these graphics is a SVG graphic, and so each cell we have a coordinate for it. And for the gene expression, we have that coordinate stored. So when you search,
So you have removed the cochlea?
Yeah.
Okay.
Yeah.
Along the cochlea, you've got a location.
Yeah.
Wow, that's cool.
And now, of course, this was done about 8 or 9 years ago, where the technology, that was the best you could do. But now actually what you can do is this technology being developed in the Brain Initiative, where you can take a slide and take a slice of the cochlea and put it on the slide. And then you can do a gene expression right there. and you know exact physical location of the cochlea where a particular gene is expressed
Code directly with the coordinate.
with the coordinates.
That's pretty cool.
So this is sort of another way where you can look at gene expression by putting in the gene, and you can sort of look at the difference. And the top, what you're seeing here is essentially two displays. This is a t-SNE map. On the right-hand side is essentially a cell type annotation. So each color represents a particular cell type in a year. And the left-hand side is showing you where is the POW4F3 gene expressed. So in this particular case, it's on the IHC's inner hair cell and OHC's the outer hair cell. So you can see that that's about the only two cell types where that POW4F3 gene is being expressed, because that has effect on your inner hair cell and outer hair cell growth. So and the rest of your cochlea, there is no gene expression for POW4F3. So this is sort of a powerful visualization tool where you can look at gene expression across time. And then you can do a few more functions. In this particular case, you can compare two different conditions and identify genes which are differentially expressed, again, graphically, and you can save those genes and then reuse them in a gene card. And that's kind of what you're showing is here. You could essentially take a little lasso-like tool and say, okay, these are the genes which are highly expressed, and I want to sort of see what are those genes, and you sort of circle around that, and that creates a little shopping cart, which you can then go and also leave as a display. This is kind of how it looks like. So if you were to draw a square lasso, it would give me a list of all the genes and what is its expression and form change. And what the other thing I want to say is, like, all these tools that we build for one project, we essentially can apply to the other ones. For instance, the gene expression analysis resource was originally built for looking at inner hair cell and outer hair cell research for Rona Otsano, but the same tool can be reapplied. So what I'm showing you is two examples, where the first example behind is where we've taken the tool without any changes and applied it to neuroscience. So Owen and Rona wrote a grant for the Brain Initiative, where they were generating single cell data, and we needed a visualization tool. So we took that data and loaded it into a gear and rebranded it, and that's called Neo Analytics. That's why the look and feel is different, but underlying it is the same technology. And then we've also taken the same technology and applied it to infectious diseases. So IGS has got a couple of grants. We're looking at infectious diseases, and we've taken that tool and loaded infectious disease RNA-seq data sets, and you can visualize that as well. So with all of these, the tools we build are not just for one custom application, but they can be reapplied in multiple different locations. All right, so I'll stop there and see if people have any questions. I'm almost coming up to 1 o'clock. I have 5 minutes left. So I have a couple of other topics. I can give you a quick overview of them, or I can stop now and take questions. It's up to you guys.
Any questions for Anup?
And have you worked with this at all?
Yeah, I worked with the PodViz, because there was like a precursor to PodViz that we developed, and then we moved on, and so often they bring me in and I look at it and I go, no, you're trying to do statistician stuff. No, I don't like this, you know.
So I have a question. I mean, this is something I've thought about for a long time, and this is the right group to ask, is in terms of graphical interaction, a user can sit down and sort of interact. Are there other competitors? Are there, I don't know the
I was
state.
wondering.
I don't know the state of the art here. Like, they go to great pains to make sure that these are web-based, but are there?
There are interfaces that have a little bit like this, maybe not exactly the same. I think your visual appearance is very attractive, to be honest. The one I'm thinking of has a slightly more maybe basic appearance in terms of color schemes and so on and so forth. But there is, it's actually based at St. Jude's Hospital for Children. And they have a large cohort of childhood cancer survivors, around 7,000 or 8,000. And you can go in pretty much along the line of what you showed us. You can go in and define a subset. You can say, yes, I want kids who had this disease at that age, and so on and so forth. And it has a little bit of that intuitive feel that your interface has. That you feel that you can sort of do it with your hands. You select them and we'll take these and so on and compare. And you can do regression analysis inside of their software, and you can do Cox proportional hazards, and you can do a logistic regression. And so it's a nice interface, and it's open source basically, or open access. You can basically just sign up for it and start using it.
And it's web-based.
And it's web-based.
Okay.
Yeah, it is web-based. Right. So yes, I think the other democratization here is to give people access to the data set.
That's why.
And they share the whole bloody thing.
Share the data
Years
set.
and years of work, and they walk the walk, I tell you, because this is what we're all saying that we should do, but we don't do it.
Well, the difficulty is, there are aspects you want to share, and then there's other things that, I know the pitfalls of our data set.
Yeah.
Right.
And I always go, oh, well, and so the original one we had done was actually looking at some trajectory-based analyses or
Yeah.
some sort of, and, they just threw things in, and I'm like, well, those are all items that are basically saying, are they right-handed? That doesn't make any sense to sit there and model right-handedness. When we're talking, or like we had, the UPWS motor has so many tremor items. Of course, you're gonna get a tremor thing, but you're not necessarily seeing something that's meaningful. And so, and those are the kinds of things that you almost sometimes, again, having,
We should nanny people or not,
Yeah.
That's one of the issues, isn't it?
yeah, or like, like I know.
The other issue is IP, obviously. People often reserve some of it to themselves because
Oh,
they say, yes, I would like to do the analysis. Have you played with the ChatGPT for data analytics?
I have.
I have too, but
Yeah.
have you?
I have not, and that's something
Okay.
interesting to sort of try out, yeah.
Yeah. Because that comes with, I think, some of the same discussions attached to it. Because suddenly you have analyses that look almost perfect on the surface now because that's how they're generated.
Right, but that's.
But what is under the hood
Or
would
you
be
can draw a
completely
nice picture
meaningless.
on anything.
Well, yeah, it's.
Well, they said like one of the early ones was they did this and they were able to predict, the number of people with malignant tumors, and that's because it picked up the code where somebody wrote malignant on the screen.
That sounds about right.
You know, like, yeah.
Turned out to be very predictive. No, that's
I
very
need
good.
to leave.
Okay,
Thank you. and
well, but.
Thank you very much. It's really interesting.
Let's give Anup a hand.